represented by that's represented by this ternary diagram. So this is actually, it's become an extremely versatile platform for loading different, mostly lipophilic payloads, but I will also be a case in which it is possible also to absorb, for example, an mRNA on the surface of them. We have actually tested different type of chitosans of varying molecular weights and degrees of acetylation. We see that regardless of the characteristics of the chitosan, it is always possible to coat these capsules with different type of chitosans, and that will determine their stability and some of their properties. Okay, so what we've been doing mostly while I was in Santiago and then carried out in Münster was to load different type of phytochemicals, mostly flavonoids. And I just refer here to some of the molecules that we've been able to load on them, like cinnamaldehyde, capsaicin, and other flavonoids such as naringenin or quercetin. I'll be mostly concentrating today on capsaicin, and I will also present some of the results with naringenin loaded nano emulsions or nano capsules. So the beauty of this platform is also that it allows us to compare nicely what is the role of coating with chitosan, because we can just have the bare pristine emulsions without any chitosan surface on them. Okay, then I just show here some of the systems that we've prepared using different chitosans of varying molecular weights or degrees of acetylation. As I said, invariably we can obtain nanocapsules, coated nanocapsules, and then as we can see also here for different payloads ranging from the ones I've mentioned before, the encapsulation efficiency is almost always very high, above 80%. And of course, nanocapsules will have invariably a highly positive zeta potential, whereas the nanoemulsions will be always invariably highly negatively zeta potential. And their size, that's another feature of these systems, that their size is very conserved. They normally oscillate around the 200 nanometers. And this is due to the process in which these emulsions are obtained by this spontaneous emulsification process. Okay, now, as I said, yeah, capsaicin has been at the focus of our attention. It is a very interesting compound. It's actually being used for pain relief or treatment, but other activities have been documented, like treatment of diabetes, urological disorders, for example, of a reactive bladder, gastric and cardiovascular diseases, cancer, appetite and satiety control. And then we've also worked and shown that it has an effect on epithelial permeability. So the receptor to which capsaicin binds, it's been well established. It's called the Trpv1 receptor. And this is actually an ion channel, a sodium and calcium ion channel. Also, interestingly, for what I will be mentioning later, it's been shown also that capsaicin, by the activation of the Trpv1 receptor, it has an effect on the sodium channel, which is very relevant in cystic fibrosis. I will be coming back to that. In previous works in the lab in Münster, we were able to show the effect of encapsulating capsaicin, for example, in modulating the effect on the reversible opening of cellular tight junctions on epithelial cells. And also, as part of those studies of Matthias Kaiser, we showed that not only that, so nano-encapsulated capsaicin has an effect on the mobility of epithelial cells with a significance that we still haven't really been able to discover, but it has a very profound effect on how cells move on an epithelial cell monolayer. Now, we've been using a platform here in the lab in Leeds to characterize not only biopolymers, but also particles. It's called asymmetric field flow fractionation. And what I'm showing here on the left panels are actually size distributions, but obtained by fractionation using this technology. And the power of this is that we can actually characterize not only the size, we can also see several populations. What I'm showing here are capsaicin-loaded nanocapsules. And then by playing with different detectors, we can determine the radius of gyration and the hydrodynamic radius, which are related but not the same. So for example, that will allow the comparison between the two, as it is shown on the lower panel on the left. That corresponds to blank nanocapsules on the black lines, or to red nanocapsules, or to capsaicin loaded on the red dots. And we can see that they are slightly different when we plot the two radius as a function of elution time in the elograms. Also, the comparison between the two radius informs about the shape of objects. In this case of these spheres, we've always seen them. Well, normally we see them as spheres on the TEM, on transmission electron microscopy. However, the ratio between them is slightly lower than what we would expect for an homogeneous sphere. And we can actually see that they're slightly elliptical. So it's a very powerful way to infer the morphology of polymers, but also of particles. Okay, now this is part of a collaborations we carried out during the Nano3 Bio project coordinated by Bruno while I was in Munster, which was a follow-up of the nanobiosaccharides project, in which we carried out this work in conjunction with Christian Gautzelani now in Hamburg. And basically, the interest here was to test the capsaicin-loaded nanocapsules against urothelial cells, due to the relevance of these capsules that could have, for example, in some urological disorders, and the fact also that the bladder is coated by a very interesting layer of epithelial cells, including, well, their own glycocalyx and mucin also. So what we did with Christian, actually, well, we prepared the capsules, labeled them with a fluorescent dye, and what I'm showing here are two cell lines, the T24 and the urotza. So you can see that the accumulation of these green vacuoles over time for different concentrations of loaded capsaicin is very different between the two cell lines. And we can see that the rate of accumulation of the vacuoles is actually significantly higher for the T24 cell line. Now, we did compare also other biophysical characteristics of these two cell lines, including the zeta potential. And that's something also we have done with bacteria. So by hypothesizing that, given that the nanocapsules are positively charged, they will electrostatically bind to cells. Then by actually measuring the surface charge of the two cell lines, we saw that the T24 do have slightly higher negative zeta potential than the urotza cells. And we can see that when comparing also the percentage of binding, it is significantly differently between the two cell lines, and both of them much significantly higher than the nanoemulsions devoid of the capsaicin coat. Then in another series of experiments, actually several enzymes were applied, and then tried to see the influence of them on the binding of the nanocapsules to the two cell lines. And then the enzymes applied were chondroitinase, hyaluronidase, and heparinase, which are enzymes that would degrade the glycocalyx of the cells. And then when comparing the two cell lines, it can be seen, for example, that there is a specificity to the different enzymes on the binding to the cell lines, connected also to the composition of the glycocalyx of the two cell lines. Not only that, in other studies, we have seen that the affinity of the nanocapsules for mucin, and these are assays carried out just interacting the nanocapsules with the purified pig gastric mucin, we can see the effect of the nanocapsules. The black bars are freshly prepared, whereas the gray ones are 24 hours afterwards. And we can see that for different ratios between mucin and the nanocapsules, there is a reversal of the zeta potential. So initially they are positive, and then as the amount of mucin passes a critical amount, they reverse and become negative. And then we also see that around the isoelectric point, that we could call it, the particles also grow in size. So there's this, we know that the nanocapsules interact with the glycocalyx, but also due to the presence of mucin, we could expect that. Now, in the case of bacteria, these are studies that we've carried out, also working together with Celina, of course. This is part of the work we did with this biosensor that Celina developed for her PhD studies, and she will be telling us all about that. So basically, we did a similar experiment as with the urothelial cells. In this case, we titrated, let's say, the bacteria with the nanocapsules. So that's what we're seeing on the left upper panel. And we can see that bacteria have also a negative surface or zeta potential, and then by increasing the ratio between nanocapsules and bacteria, we see this reversal of the zeta potential. We do not see anything of that when we add nanoemulsions, as there is no interaction, at least not reflecting on the charge. Then we were able to measure the amount, the fraction of bound nanocapsules to the bacteria by carrying out sedimentation studies. And that's the blue line on the lower panel. We can see that there is like a stoichiometric point at which the bacteria actually start not to bind any more nanocapsules. So first of all, they get fully coated, and then we start to have an excess, and then the percentage of the bound fraction starts to decrease. And these experiments were also accompanied by simulations, actually in this case only with 3 bacteria, but then by playing with the electrostatic forces and the different parameters, the size, the zeta potential, and the forces that could be modeled, it was possible to see actually a nice agreement between the experimental and the simulated data. And what we see on the center are the scanning electron micrographs of at various ratios. So we can see that a very high ratio at about in the lower right hand side panel, actually bacteria start to deaggregate. When we have too much of the nanocapsules, bacteria no longer aggregate. So controlling the aggregation of bacteria is something extremely important, extremely relevant, that has to do also with the doses that we apply. So what we've been trying to do is trying to come up with quantitative ways to understand why the nanoparticles, the nanocapsules have this kind of effects. Okay, this is part of another study carried out by Hao and Nguyen from our lab. In this case, he loaded naringenin, and it's a little bit more complex because, as I said, by having a positive surface charge, it is possible also to use layer by layer technology. And in this case, the nanocapsules are also coated by a sulfobutyl ether beta cyclodextrin at the surface. So these nanocapsules, coated nanocapsules, depending on the amount of captisol, that's the name of the cyclodextrin, we can also see a reversal of the zeta potential, as we see on the upper right-hand side corner. And we can see that as we have an excess of the sulfobutyl ester groups, then they become either negative or positive, depending on that ratio. And these capsules actually show that they have an effect on quorum sensing that Celina will be telling all about. So I will just say that depending on the coating and on the loading of the naringenin on the nanocapsules, we observe the greater significant effects on these nanocapsules, and that also has an implication on the relative antibiofilm activities. So biofilm is also an extremely important field in many fields, in food, in health, in ships, and in many fields, biofilms are a problem, but they could also be controlled. So we can see that these nanocapsules on the brown and blue bars, also significantly the loaded ones with naringenin are the ones that have greater effects on lowering the black and the brown ones have the greatest antibiofilm activity as compared, for example, to the free naringenin or the free Kaijuson on the blank nanoemulsions. Okay, now I want to now talk briefly about electrostatic self-assembly. Basically, one of the features of Kaijuson is that we can easily, nicely mix it with oppositely charged biopolymers, including, of course, nucleotides, DNA, siRNA. So then by playing with the ratios of mixing, by the molecular weights and other parameters, it's possible to obtain different type of complexes, either these simple complexes or particles, for example, if you use also TPP, or to absorb at the surface of nanocapsules. So actually, I'm gonna just go quickly over this. These are examples of Kaijuson pectin complexes that we actually use them to associate a natural extract from a plant, edible plant in Brazil that was rich in iron. And we were able to see that the complexes containing Kaijuson were able to increase the uptake of iron by CACO2 cells. Okay, now the last of my talk, I want to tell a little bit about what we've been doing for gene delivery. So basically, these are works by Marguerite Rinaldo, and actually this is just to show that when mixing Kaijuson and DNA, we would observe this reversal of the zeta potential as a function of the positive to negative ratio on the complexes. And that is also studied by gel retardation assays. We can see that depending on the ratio, it is the amount of free DNA that migrates out of the complexes. And then also studies have accompanied different degrees of acetylation. For example, when the ratio is fixed at a given pH, not much of the DNA is complex. So this is well known, actually. But what we did in this study is also in collaboration with the group of Michael Weber in Munster and Katarina Kolonko. They're focused on cystic fibrosis, and cystic fibrosis is definitely one of the very interesting diseases that could be targeted by gene delivery. In this case, we use the nanocapsules loaded with Kaijuson, but also they were coated by mRNA. And I wanted to make emphasis on mRNA because of the relevance on COVID-19 therapies also. So these capsules actually contain the capsaicin, trying to target the sodium channel that is hyper-regulated, and also to restore the cystic fibrosis chloride channel, which is also downregulated or actually is mutated in this rare disease. So, well, these are the characteristics of the capsules. So it is possible to coat them. The capsules remain stable also, stable in the medium used for doing the transfection studies. And these are the studies by doing electrophysiology measurements, by using different compounds to block the two channels, and then study the restoring of them. We can see that the blue part corresponds to those are the raw data, the raw data, sorry, and that's the restoring of the CFTR channel using nanocapsules loaded with capsaicin, also with the mRNA that restores this chloride channel. The other two bars are controls that show that's a non-targeting mRNA that shows no restoring of the channel. In the case of the sodium channel, we were expecting the sodium to be actually downregulated. However, the two cell lines tested also didn't show that much of a difference. However, there is a tendency that the loaded nanocapsules do reduce the activity of the sodium channel. So actually, there are preclinical studies based on mRNA delivery, and as I said, some of them focus on cystic fibrosis. And I just gave here 3 examples of different clinical studies that are being carried out. And this, as I said, is relevant for COVID-19, as we know, because of the mRNA vaccine. So I just refer here to a few studies currently undergoing. Inhaled Kaijuson-coated DNA vaccines, localized and systemic immune responses against SARS-CoV-2 following mucosal immunization. And I hope you will agree with me that by harnessing the mucoadhesive and the adjuvant properties of Kaijuson, probably there will be a way to develop a mucosal vaccine. There is already, for example, one company in Switzerland that it's actually advertising already Kaijuson nanoparticles for aerosol formulations for COVID-19 drugs. So with that, I want to thank you very much. I hope I didn't exceed the time. And I want to show, of course, thank here the two groups. So this is the Munster and the Leeds groups. And just before the pandemic struck, we had this summit in Cologne. We met with the groups of Leeds and Munster together. So thank you very much for your attention. And of course, thanks to all our students and who have done all of the work that I have presented, of course. Thank you very much, and the funding agencies. Thank you.
So thank you so much, Francisco, for this incredibly exciting presentation. It was really, wow, it was really exciting to see what you're working on and what you worked on. Are there any questions from the audience? Bruno, you have a question, or?
Yes, I would like to start with a question, please. Francisco, you showed us that these chitosan-coated nanocapsules can be taken up by human cells. And then you showed, and this is because they attach to the surface nicely. And then you showed that by treating the cells with different types of enzymes, you can influence this binding.
Yeah.
But you showed that in one case only one enzyme, and for the other cells different enzymes were effective. So my question is, do you think there is a specificity in the interaction between chitosan and the glycocalyx, or is it simply charge-based, and so chitosan will bind to any glycocalyx because it's negatively charged?
Yeah, that's of course, Bruno, a very, very important question, which I don't think we know the precise answer. Of course, we see that the glycocalyx influences the net charge, as we can see. Are you still seeing my slide? So we can still see that there is a slight difference in the zeta potential, but it's not, well, it's significantly different, but it's really not that big difference. I think there is a specificity, Bruno, if you ask me. As we know, also, we know that chitosan has affinity for von Willebrand factors and other aspects in other type of cells. So yeah, there must be a specificity. Now, this, Bruno, is chitosan, which is restricted at the surface of a nanocapsule, which is also may not apply to free chitosan. I mean, as we don't know really how that chitosan is really fixed at the surface of the capsules, even if there would be like specific sequences. I know that maybe there is, of course, some specificity due to given patterns, but again, there is an added level of complexity by the fact that it's actually immobilized in the surface. But this experiment says that there are differences also in the composition of the glycocalyx of the two cell lines also.
Okay.
Isn't it?
Thank you.
Thank you, Bruno.
There's one question in the chat, which is, how do you control the size of the nanoparticles coated by chitosan?
We can control it only to a very small extent. In the nanocapsules, the size is given by the process used for this spontaneous emulsification. And it's really always, so if we don't coat them with anything, they will come up invariably almost around 200 nanometers. And then, of course, if we add maybe higher molecular weight chitosan, yes, the size could be extended. Yeah.
Okay, exciting. Thank you.
And question?
Yes.
Francisco, in the work of how with the naringenin inclusion complexes with cyclodextrin, did you measure the release profile? Is a burst release or a controlled release of naringenin? Do you know?
Yes, and we did not see very, very big difference, if I'm not wrong, Celina. We normally see that the release does not explain the difference, as we've seen also with cinnamaldehyde or capsaicin. So we see that release typically is about 20% in the time course, and not much difference always between nanoemulsions and nanocapsules. But it's not a burst release. I don't have the slide here with me, but it's not burst release. It's a more controlled sustained release.
Thank you very much.
Thank you.
This is Carmen. I can make a question. I cannot turn on the camera. I don't know why. Francisco, very nice talk, as always. And I know you have been doing a very strong work on nanocapsules and also for gene delivery. I'm working also in nanocapsules for gene delivery.
I know.
For pulmonary administration, actually, my student Stefania will defend the PhD soon. So right now you are also interested or working in pulmonary delivery also, about this slide about the COVID and all that.
Yeah, we would love to be working on that. Sure. We are not doing, apart from this collaborative work with the group in Munster on cystic fibrosis, but not yet at all in animals. It's all done by in vitro work, Carmucho. So yes, we would definitely be very interested to explore collaborations in this space. Yes, we've been gaining proof of concept that yes, it's possible to deliver mRNA, active by mRNA to, I think cystic fibrosis is a good example of a rare disease in which gene therapy probably will, of course, not vaccination, of course, right, as we are already seeing gene therapy and huge millions.
No, Francisco, I tell this because I hope we can talk in the future. I think it will be very interesting, the pulmonary delivery of some vaccines also for COVID, for example. People are exploring for nasal administration, that of course is easier, but at least I think I have the technology for pulmonary administration to be able to put in the lungs. So I think it's very interesting, and it could be nice to talk more.
Thank you, Carmucha. We'll be looking forward to talk, of course, and very open, very happy to try to collaborate again, of course. Thank you.
And it's nice to remember your stay in Santiago. As Bruno told, you are a traveler, but we were lucky that one of the steps of your life was Santiago de Compostela. And also Bruno's meeting about nanobiosaccharides. That was very nice. I always remember that meeting and also the funny things. Okay, thank you.
Thank you, Carmucha. Thank you.
Okay, thanks you all for the questions, and thanks Francisco again for this very exciting presentation. Thank you so much for contributing today.
I thank you, Gregor. Thank you all. Thank you.
And in the interest of time, we just continue with Celina.
Yeah.
Celina Vila. It's a pleasure to introduce you, Celina. Celina graduated in biology at the University of A Coruña and recently defended her PhD, her doctoral thesis at the University of Santiago de Compostela. So all congrats to that, Celina. And yeah, Celina performed large parts of her doctoral project between the NanoBioFar group in Santiago de Compostela and the group of Francisco, still back in Münster. Is that correct?
It's correct.
Okay, great. Yeah, and in your doctoral thesis apparently evolved around the hypothesis that bacterial quorum sensing can be explained as a percolation process in which connectivity is more important than the sheer density in a bacterial population, which apparently can have unforeseen implications in understanding cell-to-cell communication mechanisms. So I'm really exciting. When I read your CV, I was like, oh yeah, that's a thing. Really exciting. And Celina, we're looking forward to your presentation.
Thank you very much for the kind introduction, Gregor, and for the invitation to be here today. It's a pleasure. I will share now my screen.
Perfect.
Hope it's okay?
Yeah, perfect, all working.
Okay, so today I'm not going to talk about percolation. Due to time limitations, I will focus on the application of dually cross-linked Chitosan nanoparticles to block quorum sensing and target bacterial growth. Thanks, Francisco, for the very nice general overlook on the topic. It's gonna ease my presentation a lot, I think. Ay, espera, no me va. A ver si va ahora. So let me close this. Okay. So during this presentation, I will give an introduction on quorum sensing and quorum sensing interference strategies before going to my experimental part on the use of Chitosan nanoparticles. So quorum sensing are cell-to-cell communication mechanisms via secretion and sensing of chemical signals or autoinducers. And autoinducers vary among gram-positive and gram-negative bacteria, but the family of the acyl homoserine lactones or AHLs are widespread among gram-negative bacteria. Here you can see a general structure of these molecules. And AHL-inducible LUX-R regulatory systems of the kind depicted here by these two talking bacteria control a wide variety of bacterial traits of high impact, not only in human health, but also in agriculture and aquaculture, among others. And these include the formation of biofilms, pathogenesis, production of virulence factors, among others. So it's not surprising that strategies aimed to interfere with quorum sensing are considered nowadays as a powerful tool to develop alternatives to classical antibiotics in the context of spreading of antimicrobial resistances. And these strategies are also known as the general name of quorum quenching strategies, can fight pathogenic bacteria directed to many quorum sensing targets. I'm not going to explain in depth all these targets, but I will focus specifically in the use of nanoparticle-based quorum sensing inhibition. Nanoparticles, due to their unique physicochemical characteristics and their surface to volume ratio, have unique properties that combine a broad spectrum antimicrobial or also anti-quorum sensing activities, and they can also be functionalized to bear a number of functionalities, or also to incorporate cargoes like classical antimicrobials or anti-quorum sensing compounds that can be more effective towards resistant pathogens. This makes nanoparticles excellent candidates to develop new generation antimicrobials. And a main part of my thesis was focused in the use of Chitosan-based polymeric nanoparticles intended to interfere with quorum sensing. On the one hand, because Chitosan is a key material both in my groups in the University of Santiago and the University of Munster to develop nanosystems, and also because Chitosan polymers and Chitosan-based micro and nanoparticles are amply recognized to possess a broad spectrum antimicrobial activity, including towards antibiotic-resistant pathogens. And the antimicrobial mode of action is not yet of Chitosan is not yet fully understood, but at least in gram-negative bacteria, two main mechanisms are undercover, which the first one involves Chitosan electrostatic interaction with the lipopolysaccharide on the outer membrane of gram-negative bacteria, and another mechanism involves Chitosan interaction with outer membrane proteins of the OMP-A family. And altogether, these two mechanisms seem to disturb and alter the permeability of the outer membrane. And recently, a paper has also related Chitosan activity to reprogramming of cell metabolism in response to stress. However, the applications of Chitosan for anti-quorum sensing strategies is still a research area in its infancy. And in this table, I summarize the application of Chitosan and Chitosan-based materials to interfere with quorum sensing, in which our group in Munster has significantly contributed, and all these systems have been already cited by Francisco in his previous talk. As you can see, these works are still growing. I couldn't include, for example, the new very interesting paper of how Guyan. So in general, Chitosan and Chitosan-based materials have been proven to interfere with quorum sensing traits such as biofilm formation or production of virulence factors in synergy with classical antibiotics or with known quorum quenching compounds. But only a couple of studies have related the intrinsic quorum quenching activity of Chitosan. And in this regard, the work that I have developed during my thesis has contributed to understand the bioactivity of Chitosan nanoparticles in the context of an E. coli-based quorum sensing biosensor. And the results of these investigations have been published in 2 papers in the Journal of Colloid and Interface Science. So I will start by describing the E. coli biosensor that we use in our studies. Next, I will explain the 2 fabrication methods that we use to obtain Chitosan nanoparticles by means of ionic and covalent cross-linking, and the impact of genipin cross-linking on the ultrastructure and colloidal properties of Chitosan nanoparticles. And at the end, I will focus on the effect of Chitosan nanoparticles on the growth and quorum sensing of the E. coli biosensor. So the E. coli biosensor cells carry a leucide-deficient genetic device based on the lux operon of a Vibrio fischeri. This means that the biosensor cells are not able to synthesize the AHL, but they respond to exogenously added AHL in a predictable manner by expressing GFP under LuxR AHL control. This illustration shows a typical biosensor assay performed in our lab, where we add AHL or AHL and Chitosan nanoparticles at different at the specific concentrations on biosensor cultures, and we monitor fluorescence and optical density over time. Here you can see the typical quorum sensing response of the biosensor at increasing AHL concentrations. We measure the response as a density normalized fluorescence, and typically the response increases up, grow up to a maximum level whose magnitude is determined by the AHL concentration. And I want you to keep this figure in mind, because this is the kind of response we want to inhibit with our nanoparticles. So we have developed 3 types of nanoparticles, namely IC or ionically cross-linked nanoparticles, CC genipin co-cross-linked nanoparticles, and PC genipin pre-cross-linked nanoparticles, prepared by 2 fabrication methods that are represented here in this scheme. The starting material was a commercial Chitosan of high purity in the hydrochloride cell form with an intermediate molecular weight and a degree of acetylation of 14%. Method 1 is initiated by ionic gelation of Chitosan with TPP to render the ionically cross-linked nanoparticles by the method developed in our group in Santiago, and followed by genipin covalent co-cross-linking to render the co-cross-linked nanoparticles. And method 2 is an inverted version, and it starts by a covalent pre-cross-linking of Chitosan with genipin to obtain a pre-gel Chitosan that is further cross-linked with TPP, ionically cross-linked with TPP to render the pre-cross-linked nanoparticles. And this is a novel method that we have developed in our lab in Münster. The genipin cross-linking reaction of Chitosan with genipin involves the nucleophilic attack of a glucosamine residue of Chitosan on the heterocyclic ring of genipin, and a secondary amide link formation with another glucosamine residue of Chitosan. And we have observed that genipin cross-linking has an impact on the ultrastructure of both genipin co-cross-linked and genipin pre-cross-linked nanoparticles. So starting with method 1, genipin co-cross-linking of Chitosan TPP nanoparticles stabilizes a pre-existing but weak core shell structure. We follow the evolution of the cross-linking reaction of ionically cross-linked nanoparticles with genipin at increasing genipin to chitosan mass ratios and at different incubation times at 37 degrees by SACS. And we feed the SACS data to a core shell model and calculated the shell thickness in the cases of a success of fitting. And as we can observe in this diagram, ionically cross-linked nanoparticles in the absence of genipin showed a shell of about 50 Armstrongs at early times of incubation, but the dashed lines show that the data was no longer could not be longer fitted to the core shell model at longer incubation times, which implies that the surface structure of the ionically cross-linked nanoparticles is very unstable at upon incubation at 37 degrees. And the same trend was observed for very low amounts of genipin, whereas for increasing genipin to chitosan mass ratios, the original shell was not only maintained, but increased in thickness at longer incubation times. Coming to method 2, as a reminder, started with chitosan and a premix of chitosan with genipin. Chitosan pre-cross-linking with genipin induced a sol-gel transition. So we followed the cross-linking reaction of chitosan and genipin by dynamic light scattering, and we observed that the correlation coefficient decayed with time of incubation, and so did also the maximum values. And when we plotted the areas under the curve of these correlograms over time, we obtained a monotonic decay, which was consistent with the transition from a liquid-like to a gel-like structure. And we estimated the critical gelation time, both by the dynamic light scattering and rheology, and estimated these values between 11 and 18 hours of incubation of the mixes. So the effect of genipin cross-linking on the outer structure of co-cross-linked and pre-cross-linked nanoparticles also translated to an impact on the colloidal properties of these nanoparticles. And we observed that genipin cross-linking avoided the tendency of chitosan nanoparticles to aggregate after isolation. So nanoparticle isolation upon centrifugation and resuspension is a critical step during the fabrication of chitosan TPP nanoparticles. And we observed that ionically, in method 1, that ionically cross-linked nanoparticles after isolation suffer a 3-time increase of size compared to the non-isolated counterparts. Whereas the genipin co-cross-linked nanoparticles after isolation maintain the original size of the ionically cross-linked batch. This trend was also observed in the polydispersity of these nanoparticles. And in method 2, genipin pre-cross-linked nanoparticles maintain the original size also after isolation. And moreover, genipin cross-linking ensured colloidal stability of both genipin co-cross-linked and pre-cross-linked nanoparticles in the microbiological medium used with our biosensor, while the ionically cross-linked nanoparticles were very unstable and aggregated in this medium. Lastly, we investigated Lastly, we investigated the effect of the 3 types of nanoparticles on the growth and quorum sensing of the E. coli biosensor. Here you can see the bar diagram showing the response of the biosensor upon treatment with a increase in concentrations of nanoparticles relative to control cells. And strikingly, ionically cross-linked nanoparticles, independently of the dose, promoted a very drastic reduction of the response of the biosensor. And despite their improved colloidal properties, the genipin co-cross-linked nanoparticles did not show such an apparent effect, whereas genipin pre-cross-linked nanoparticles at medium to high concentrations promoted a very strong reduction of the quorum sensing response of the biosensor in a manner similar to the ionically cross-linked nanoparticles. And due to the combination of the optimal colloidal properties and quorum quenching activity of the genipin pre-cross-linked nanoparticles for experiments that I will show from now on, I will focus on this kind on this type only. On this type of nanoparticles. So here you can see the raw fluorescence plots, which evidence in a better way the mode of action of the nanoparticles. In the absence of nanoparticles, the biosensor displays a biphasic response with an initial fluorescence burst, represented here in the first box, followed by a steady increase of fluorescence up to a maximum. This is the typical response that I showed before. When we apply the nanoparticles, most nanoparticle concentrations promoted a dilution of fluorescence following the initial burst. That is here represented by the arrow. And most nanoparticle concentrations promoted the appearance of shoulders on the OD traces. They are also here shown by the arrows. We don't know what these anomalies on the OD reads mean, but we know that there are aggregation events, as Francisco previously explained, related to the application of Chitosan-based nanomaterials on bacteria, specifically on this biosensor. And despite the high variability among experiments, in the two papers, we have provided all treatment replicates to characterize the presence of these anomalies. And in many cases, we observe that after the OD anomaly, the OD trend comes back to the normal trend. So we don't know what exactly happens after these shoulders appear, but we have proposed a very interesting mode of action of the nanoparticles that is consistent with the existence of the stoichiometric ratio of nanoparticle to bacterium that Francisco already mentioned for Chitosan-based nanocapsules that was very well characterized by us with the nano capsules. As Francisco mentioned, the stoichiometric ratio is the point when the negative potential of the bacterial envelope is counteracted by the positive charge of the nanoparticles. And we inspection all treatment replicates in the search of these OD anomalies. An example of a representative experiment is shown in this slide. The first panel represents the untreated cells and is again represented by the orange traces in the other panels, and B2E panels represent increasing concentrations of PZ nanoparticles, and the black to gray traces represent independent treatment replicates within a single experiment. We consistently mapped the timing of appearance of the OD anomalies. For all experiments, we have found that the cell density at which the stoichiometric ratio is established displays clear dose dependence relative to nanoparticle concentration, as we show here in this second graph. And this behavior implies that we can anticipate the growth phase at which the stoichiometric ratio will appear by simply varying the nanoparticle concentration. We believe that this behavior of Chitosan nanoparticles could be exploited in contexts where targeting specific cell densities is of interest. And this doesn't just limit to the pre-cross-linked nanoparticles, but as Francisco mentioned, applies to even polymeric chitosan or chitosan-based nanocapsules, because we consistently observe in our lab this kind of behavior that we believe it deserves further attention for very promising applications. And these applications could, for example, include fermentation processes or waste management, which rely on directed flocculation of bacteria. And in this context, the use of biopolymeric eco-friendly materials in opposition to synthetic or metallic ones are of particular interest. And chitosan nanoparticles can also be engineered to carry and deliver bioactive molecules locally, for example, antimicrobial compounds or anti-quorum sensing compounds, such as the naringenin that Francisco just mentioned, or cinnamaldehyde, for example, a lot of examples that we have in our lab, and a wide battery of compounds that we can include. The applications of chitosan nanoparticles to target bacterial populations can also be applied to separate food contaminants and pathogens. And this behavior can also have effect on the ability of bacteria to form biofilms or to respond to antibiotic phages. And this context is of particular interest to fight antibiotic-resistant pathogens. And lastly, it was recently proposed that chitosan can compete with aggregation processes of pathogenic bacteria mediated by endogenous polymers in infection sites. For example, in the context of wound infections caused by Pseudomonas aeruginosa, or also in the context of lung infections, such as cystic fibrosis. And thus we propose that our chitosan nanoparticles could be candidates to interfere with this kind of processes in infection contexts. Of special relevance, as Carmen Remuñán already said, is the lung context, and where she already worked with lipid nanoparticles. So we think chitosan nanoparticles could be a next candidate to explore. So now I would like to highlight the more relevant conclusions of the work presented here. So we developed an alternative method to covalently modify chitosan nanoparticles with geniping as a strategy to improve their colloidal properties while maintaining their quorum quenching activity. The apparent growth inhibition of chitosan nanoparticles on biosensor cultures was consistent with the existence of a stoichiometric ratio of nanoparticle to bacterium during cell growth. And the establishment of this stoichiometric ratio displayed clear dose dependence on bacterial density, and its timing could be controlled, implying that these nanoparticles could be ideal to target bacterial populations at specific cell densities. And now I want to thank the European Chitin Society, especially Bruno and Gregor, for the invitation to participate in this very nice webinar, to my PhD supervisors, Carmen Remuñán, Francisco, and Anton, that are here also today online, and a special mention to the co-authors of the papers that I presented here today, David Lohan, Lea Hembach, and Julius Netza, and to all of you for your attention. Thank you very much.
Thank you so much, Celina. Really exciting talk. Glad that you found time to present today. Are there any questions from the audience? A lot of emoji claps. Okay, Karen.
Okay, may I? Okay, thank you. Congratulations to both speakers. Of course, Francisco, I didn't mention you, but you are in my heart, as you know. And I have a question for Celina. This maybe also to Francisco, this stoichiometric ratio that you mentioned is based mainly of a factor of size, meaning the specific surface area difference between the bacteria and the nanoparticles. So in this way, it could be better calculated, let's say, in advance. Or is there any other mechanism involved that may be more specific? Okay.
It's a very, very interesting question. Thank you very much. So we do know that the mechanism involved is electrostatic interaction of chitosan with the negatively charged outer membrane of bacteria. And I think the next step would be to specifically address this question by applying different nanomaterials and also nanoparticles of different sizes to explore where if there is an effect of size. I don't know, but we also applied in many cases. So we also observed the same anomalies with OD anomalies with polymeric chitosan. So I'm not very sure if size is a critical factor in this behavior. We believe that this behavior can be is a general mechanism of chitosan-based materials, but we don't know at the moment details. Also, another issue would be to address the effect of a degree of acetylation or pattern of acetylation on this behavior, and I think it will be a focus of further investigations in the future, for sure.
Okay, probably you're right. It's not that, but it was evoked to me by your slides and also by the slides of Francisco, when you see all the bacteria covered with small nanoparticles. But of course, also the range of dimension is quite different, so it's okay not to be that sure. Thank you very much. Thank you.
If I might just quickly comment here, yes, definitely the size, Carla, is important. We've tried to model that in the simulations we did. We just assumed that all the capsules were of the same size, which of course is not a reality. So we know they are highly polydispersed, but we think that the size plays a role because that's actually determines how many particles will actually bind at the surface. And then if you consider polydispersity, of course, larger particles together with the smaller ones will actually fill out all the void spaces there. So this is very relevant in paints, for example. It's not interested to have all the particles of the same size, but to have polydispersity, and then the coating is much more efficient. But that's definitely something we need to investigate further.
Okay, if there are no more questions, are there any more questions? If not, oh, Bruno, you have a question?
If I may, Celina, you already answered many of my questions during the defense, but I still have another one. You showed these two types in which you can prepare these cross-linked particles. And you also very nicely showed that both types of cross-linking, either after formation of the particles or before the formation of the particles, will improve the stability of the particles during the processing. But for some reason, only the pre-cross-linked ones maintained the quorum quenching activity. Do you have a possible explanation, a hypothesis for why this is?
So I think the inner structure of the nanoparticles, the co-cross-linked and the pre-cross-linked is very different, because in the pre-cross-linked nanoparticles you have like a homogeneous pre-gel and then a further cross-linking with TPP, and we believe that it has like a dense core and a more lax surface, whereas the co-cross-linked nanoparticles are ionically cross-linked with TPP and with these soft nanogels, and then they seem to have like a rigid surface. And I don't know if this is the reason why the bioactivity is so different, but we don't have any other clue about that. We just found it strikingly, this big difference between the two types. And I also believe that we have to really characterize this in a better way. So at the moment, I don't have the details on the bioactivity.
But I think it's quite reasonable to assume that the more extended shell, where the chitosan is probably less densely packed, allows better interact with quorum sensing.
I also think this might have a big impact. We will go further into that as well.
Good. Okay. Thanks.
Okay. So with that,
I would like just to, and Carmen again, sorry, I cannot turn on the hand or to turn on the camera. Just to congratulate Celina. I don't have questions. I already made them a few days ago, and congratulate her for the presentation, for her thesis book. That was a very good work, mostly due to Francisco Goycoolea and Anton. And just to say again, that was the best presentation of PhD never seen in our group. So congratulations. And also thanks to Bruno for attending and nothing else.
Thank you very much, Carmen.
Okay.
Thank you very much.
Great, then thanks so much again to Francisco and Celina for this great presentations. It was a really exciting topic that was covered today. Thanks everyone for taking part at this webinar. We continue on 31st of August with our next webinar, which includes a contribution of Marcin Strusik about the medical application of chitosan. So kind of leaning on to the applications of what was discussed today. And yeah, thanks everyone. Have a good day and see you next month.
Thank you very much.
Thank you very much, Gregor and Bruno. Yeah, very nice.